Hi, and welcome to this run-through for the AQA A-Level Biology Paper 2 from 2019. So if you're doing this in 2024, 2019 is a good paper to practice on because it's recent, but it's not so recent. So hopefully some of the things that come up in this 2019 paper may also appear in the 2024 paper. So let's get into it. All right, question 1. Okay, so question 1 is about succession. Succession occurs in natural ecosystems. Describe and explain how succession occurs. Quite a nice, easy question to start, just kind of a recall question. I put this picture over here just to go through it with you. So succession starts when we have bare rock. This is primary succession when we don't have any soil present. Lichens colonize that bare rock. And some small mosses as well. And as they die and then decompose, they start to build up soil. That makes the environment less hostile for other organisms. So other plants, secondary colonizers, come in, and this process keeps going with these plants dying, decomposing, building up more soil. Biodiversity increases, biomass increases, and ultimately we reach a climax community. Okay, let's check the mark scheme to see what exactly you need to say. So here it is. Colonization by pioneer species. Pioneer species. They change the environment. They make it less hostile for other new species. Notice the underlining, less hostile as a keyword for new species. This bit you just need to say that changes the environment. You could either say it becomes less hostile or you could say it becomes more suitable. It's two sides of the same coin. Increase in biodiversity to a climax community. Okay. Be aware that sometimes, depending on the type of question, you may get questions that ask you to kind of describe the changes in biodiversity. And in some questions, you'd probably be presented with data. You might have some sort of graph that looks a bit like this, with biodiversity on the y-axis and time on the x-axis. In some questions, you might get an indication that biodiversity increases, but then comes down a little bit in the climax community, because occasionally, depends on the ecosystem, biodiversity can be higher in those kind of transitional moments, in this area sometimes. Okay, on different questions. Let's keep going. Okay, so we've got energy flow here. Figure 1 shows percentages of energy transferred from sunlight to a zebra in a grassland ecosystem. So let's just kind of analyze it first. We've got the sunlight coming in. Only 2.1% gets into the vegetation, so that's really not a lot. And then 12.7% gets into the zebra. Okay, so that's 2.1% of the light energy is stored as biomass in the vegetation. And then only 12.7% of what is stored in the vegetation makes it into the zebra. And then we have these various losses. 36% respired, 58.2% feces and urine, only 5.7% new tissue. Wow. Okay, use figure 1 to calculate the percentage of sunlight energy that would be transferred into the feces and urine of a zebra. Okay, so that's important. Because I just assumed that the question was going to ask me how much makes it into the new tissue. So I almost just went ahead and calculated the wrong thing. It's into the feces and urine, so we want to look at this number here. Okay, so we want to do, for 100% of the sunlight, 2.1% into the vegetation. So we want to do 2.1% times 12.7% times 58.2%. Now we're going to work in decimals, so that would be 0.021 times 0.127 times 0.582. Let's work that out. 0.001552. Now we want to give this as a percentage to 3 significant figures, so what we've got to do is we've got to move the decimal place here to turn it from a decimal into a percent. We've got to move it 1, 2. So it's actually 0.155, and that's the 3SF. 0.155. Let's check. Is that right? Yes. Yes, it is. Okay, good. All right, let's keep going. In this ecosystem, the net productivity of the vegetation is 24,525 kilojoules per meter squared per year. Use this information and figure 1 to calculate the energy stored in new tissues of the zebra in kilojoules per meter squared per year. Okay, so what figure are we given? So the net productivity of the vegetation, that's this number. So we've got that is 24,525. And we want to work out how much goes into the new tissues of the zebra. So we need to times it by the decimal value, which is 0.127, and then we need to times it by 0.057. That times that should give us this. Let's work it out. So I've got 177.5. Doesn't tell me how many SF it wants, so I'll probably just leave it as 178 kilojoules per meter squared per year. Let's check the mark scheme. Oop, wrong one, that one. Okay, 178 or 17, either of these would be fine. Okay, let's keep going. Question 1 done. Okay, this question, sickle cell disease. Sickle cell disease is a group of inherited disorders. People with sickle cell disease have sickle-shaped red blood cells, a single base substitution mutation. I think let's highlight key bits here. Single base substitution mutation can cause one type of sickle cell disease. This mutation causes a change in the structure of the beta polypeptide chains in hemoglobin. These sorts of questions come up again and again. This is a question looking at how a mutation can cause a change to protein structure. And we just really have to flesh it out. We need to say that a single base mutation can change the codon, can change the amino acid to give it different properties. That will change the primary structure, change the secondary structure, and then change the tertiary structure, and then the quaternary structure of this protein. You basically just need to describe that. Now I wanted to give you a little bit of extra info on this. This is kind of going a little bit almost beyond what you need, but this sort of thing does come up repeatedly, so I just wanted to show you this real quick. So this is a schematic of the hemoglobin molecule. We've got 2 alpha subunits, I'll highlight them yellow here, and 2 beta subunits, I'll highlight them green here. Okay, 2 alphas and 2 betas, quaternary protein. What actually happens, if you want to go further, this could be something useful like post-spec information for an essay maybe, is that there's a 1 base substitution that brings in a valine. Now this valine amino acid there is hydrophobic. A hydrophobic amino acid means that it will kind of bond to, via hydrophobic interactions, not bond, but interact with, this little hydrophobic pocket on another hemoglobin. So we get this hydrophobic interaction. So 2 hemoglobin quaternary structure polypeptides kind of interacting together. And this actually forms a chain like this. So we get this massive polymer or fiber of hemoglobin. So the hemoglobin becomes not soluble, but fibrous. That's terrible because it makes these long chains or fibers within the red blood cell. So normal red blood cells flowing smoothly through capillaries, but if we get these sickled cells, which contain these long fibers of hemoglobin kind of pushing out on them, then we get these clogs, and blood clots forming in the capillaries. Okay, that's the problem. Okay, let's just look at the mark scheme answer. What words you need to say, what keywords you need to say. And this answer is something to memorize, basically. You get the change in the amino acid and the primary sequence, alters the tertiary structure. Interesting. This, I wouldn't have thought of this. I've lost the mark here. Change in hydrogen ionic disulfide bonds. I wonder if you would get except change in hydrophilic and hydrophobic interactions, because this is actually wrong. Change in hydrogen ionic disulfide bonds. It isn't a change in any of those. It's a change in the hydrophilic and hydrophobic properties of the amino acid. So that's interesting. I guess for AQA, these are the types of bond that you want to remember. Hydrogen ionic disulfide bonds. So you could have mentioned any of those. But if you want to talk about this specific mutation for post-spec essay, it is actually a hydrophobic amino acid that is incorporated instead of a hydrophilic one. Okay, let's keep going. Okay, here's a fun word to say. Hematopoietic. Hematopoietic stem cell transplantation, HSCT, is a long-term treatment for sickle cell disease. In HSCT, the patient receives stem cells, let's look at this, receives stem cells from a bone marrow of a person who does not have SCD. So remember, SCD is a genetic disease, so the stem cells coming from a person who doesn't have it don't have that faulty gene, don't have that sickle cell hemoglobin gene. The donor is often the patient's brother or sister. We should also be thinking, okay, why is that? It would be to minimize the chance of the bone marrow being rejected, get a tissue match. Before the treatment starts, the patient's faulty bone marrow cells have to be destroyed. You should also be thinking, why is that? Well, because we don't want any new sickle cells, like red blood cell sickle cell version, being made by the person's bone marrow cells. We want to kill the person's bone marrow and replace it with someone else's bone marrow. So if we kind of explain it, then that's how we get the marks. But I wanted to show you this picture as well, just a little go further on this concept of a stem cell. This is the hematopoietic stem cell that can differentiate into lots of different cell types. The ones that we're looking for in this specific disease are the red blood cells, the erythrocytes, but stem bone marrow transplants can also be used to treat leukemia, for example. Leukemia is often when you get like too many lymphocytes being made that kind of just target the body. So we might also want to use a stem cell transplant to kind of produce other cells that can be produced by this very useful stem cell. Okay, so I think the explanation of how HSCT is an effective long-term treatment, I think we need to say that bone marrow cells are first destroyed so that no new red blood cells with the sickle cell mutation are produced. And then the bone marrow of the person does not have SCD, contains the normal hemoglobin gene to produce normal hemoglobin and normal red blood cells. And the patient's brother or sister is often the donor to minimize chance of immune rejection. And then we also need to say that the stem cells differentiate to produce red blood cells with the correct hemoglobin. Okay, so healthy red blood cells or normal polypeptide would be credited for the first mark. Second mark is talking about the fact that we want to kill the bone marrow that exists in the person already, so that's no new faulty blood cells are produced. And okay, look, again, little language issues that I might have missed here. I think I did miss. So we need to say divide. Stem cells divide to produce red blood cells. We don't actually... Oh, this, I would have got this mark, actually. Less chance of rejection from brother and sister. But this language here I used was not what they were looking for. We need to say divide or replicate. It's not the same as differentiate. They do both. The stem cells differentiate and divide and replicate. They do all of those. But this question was looking for the production, the replication, not so much the differentiation. Okay. A new long-term treatment for SCD involves the use of gene therapy. Here are some of the stages. Okay, so let's look through it. Okay, so first we get the functional gene, so that's without the mutation, for the beta polypeptide of hemoglobin. Many copies. Okay, so maybe we're doing in vivo cloning or in vitro cloning, probably using maybe PCR possibly. Could be. The beta polypeptide gene is inserted into a virus, so this virus is going to be a vector, I presume. Yeah, so the virus carries the gene into stem cells. Okay, so that's sample of faulty stem cells removed from the child's bone marrow. Okay, so they're their own cells, their own stem cells, but we just replace the defective gene with a good copy of the gene. And then we put this genetically modified stem cells back into the child. Okay, sounds very sensible. Some scientists have concluded that this method of gene therapy will be a more effective long-term treatment of SCD than HSCT. Use all the information provided to evaluate this conclusion. All the information provided, so from figure 2 and figure 1, that's pretty much all the information. Okay, so what I'm thinking is 3 marks. So the key thing is that the stem cells are from the patient, so they will be an exact tissue match. There'll be no chance of the stem cells being rejected or targeted by the person's immune system. And also that person will not have to take immunosuppressant drugs. So for any organ transplant, if it wasn't their own stem cells, they'd have to take immunosuppressant drugs over the long term. That's like a pro. I'm trying to think what's a negative. However, we could say that there may still be, okay, so in this child, they may still be producing some defective sickle cells, because it doesn't look like from this process that we actually kill their bone marrow cells, and we can't be 100% sure if the correct gene has been inserted into every single stem cell. So maybe some of the stem cells might still be producing the old type of hemoglobin. That's what I'm guessing. I think that would probably be enough if we explain both kind of sides of the coin. Let's have a look at the mark scheme and see if there was anything else that was possible. Okay, yeah, so that's what we just said. So no destruction of bone marrow or no destruction of stem cells. Oh, that's a 4. That's a 4 thing. I would almost thought that would be an against thing. Donors are not required. This is key. Less or no chance of rejection. Okay, so these are all 4. And these down here are against. Okay, so yeah, so the flip side of this first thing, you could argue it's a positive. They don't have to take a bone marrow killing treatment. But the flip side of that is that, doing green, the sickle cell, faulty sickle cells are still produced. This is interesting. Immune response against genetically modified virus. Now, can I just quickly go back to the flow chart, the flow diagram? Because, okay, yeah. No. This is so silly. Sometimes AQA makes... Okay. This is actually incorrect, AQA, because the stem cells are removed from the child. They are in a sample. They're in a petri dish. And the transfection, where the virus infects the stem cells, is happening in vitro. Okay, it's happening in a dish. And then it's being injected back into the person in vivo. So I don't agree with this mark scheme point, even though it is there. The immune response against the genetically modified virus shouldn't really happen because the virus is only in the petri dish, not in the person. This is a bit of an easy one to just give a go. Long-term effect not known. So with any gene therapy, sometimes there's a chance that when you put the new gene in, we don't always know where the gene goes, whether it goes exactly the right place we want to put it, or whether it maybe goes into a different chromosome than when we were expecting. That might cause problems with maybe the cell cycle. It might cause uncontrolled cell division, possibly. So that's kind of what they're angling for, could cause side effects. But it looks like there's more pluses than negatives. Okay, on to question 3. It says, there's a lot of stuff here, so we're going to have to really analyze this method. When you get a big method like this, it's likely that you're going to have to maybe improve it, find flaws in it, so let's really make sure that we kind of process it all. The student investigated the effects of IAA on the growth of oat seedlings, young plants. Okay, so what do they do? First of all, they remove the shoot tip from each seedling and cut out a 10mm length of shoot. They placed 10 lengths of shoot into each of 5 petri dishes. So I guess that's, so they need 50 bits of shoot. Yeah, I'm just going to write that down there. And that's like repetition, isn't it? 10 lengths. So we're going to probably calculate a mean, I'd imagine, from the 10. Added to each petri dish an identical volume of 5% glucose solution. So that's a control variable. We've got same volume and 5% glucose solution. Added to each petri dish 40 centimeters cubed of a different concentration of IAA solution. So that's the control variable, is the 40 cm cubed. And then the independent variable, the thing we're changing, is the concentration. So that's the CV and an IV. Left the petri dishes at 20 degrees C. Again, control variable. In the dark, control variable, with their lids on for 5 days. Lots of control variables here. This is looking like a pretty good method. Removed the shoots after 5 days and measured them. Determined the mean change in length of shoot at each concentration of IAA. Okay, all right, so here are the results. First thing to notice is that this is like, we're working with powers of 10. 10 to the minus 5, 10 to the minus 3, 10 to the minus 1, 1, and 10. This is like 10 to the 0, isn't it? 10 to the 0, 10 to the minus 1, 10 to the 0, 10 to the 1, 10 to the minus 1, minus 3, minus 5. So this is a logarithmic scale. If we plot it on a graph, it's going to, the x-axis is going to be logarithmic. Mean change in length of shoot, no effect, and then this is just basically increasing, getting more and more increasing as we increase the concentration. I would say there's very little effect in this range, 10 to the minus 5 and 10 to the minus 3, and then there's a bit more of an effect after this step up to 10 to the minus 1. Okay, first of all, explain why the student removed the shoot tip from each seedling. Well, the shoot tip is the place where auxin is normally produced. I say auxin, IAA. Auxin is a type of IAA. No, IAA is a type of auxin, but I'm just going to say auxin because it's easier to say. So auxin is normally produced in the shoot tip of a growing seedling, and it kind of diffuses down, so that the auxin is kind of produced in the shoot tip and it diffuses down. So in this investigation, we're looking at the effect of auxin. So we want to remove any auxin that is produced from the plant. So we have to remove the tip, cut it off, so that it's only the auxin on the petri dish that is having the effect. So let's see if that gets us the marks. So 1 mark for saying that the tip is what produces IAA. It does say except auxin, but we should probably just write IAA. Tip produces IAA, affects concentration of IAA. Yeah, I think we would... 2 and 4, ignore affects results, except affects independent variable. I think I think you would probably get this if you said we want to make sure that the concentration of auxin is as specified on the petri dish and not from any extra auxin that is produced. Perhaps. But a perfect answer from the mark scheme would be the tip produces IAA. This would affect the concentration of IAA, causing mitosis that would affect the growth of the shoot, or the elongation of the shoot. Yeah, okay. Let's keep going. .2. Explain why the student added glucose solution to each Petri dish. So this is just for respiration, to provide a source of energy. And we might also just say that because the shoot tips are in the dark, there's going to be no photosynthesis producing glucose for the plants themselves. That should probably do it. Let's see what the mark scheme says. For respiration, to provide a source of energy. Yep. And is there anything about... No, it says ignore photosynthesis, so no marks for that on this particular question. But yes, it is true. The plants would not be making glucose for themselves. Why would you have to keep the lid on a Petri dish? This is more of like a practical consideration. If you can just kind of imagine what's going on in the Petri dish and consider the timeline. They were left for 5 days, the Petri dishes. So if we didn't have the lids on after 5 days, the Petri dishes would dry out, water would evaporate, and the seedlings would probably die. So I think that's what we need to say. Mark scheme. Okay, I'd get 1 mark for that. So to prevent or reduce evaporation, this would kill the seedlings, to be honest, after 5 days. But this question is wanting you to focus on the concentration of auxin itself. So when water would be evaporating, the liquid would be concentrating. So the IAA concentration would be increasing, and therefore that would kind of mess up the experiment. So evaporation would alter the concentration of IAA, or you could have just say reducing evaporation would alter the water potential of the Petri dish, potentially harming the seedlings. Okay, 3.4. Describe and explain the results shown in Table 1 and suggest how the results might have differed if lengths of root had been used. Okay, so it's 3 marks. So I am guessing that we've got 1 mark for describe, 1 mark for explain, and then 1 mark for suggest. This is a triple command word question. Describe, explain, suggest. Right, so first of all, describe the results. So I think we need to say that that there is a general increase with increasing concentration of IAA. There is an increasing change in length, and I would add to that there's very little change. Well, I'd probably just say there is 0 change with 10 to the minus 5 and very little change up until 10 to the minus 3, and then after 10 to the minus 1 we have a bit more of an increase. So that's my describe. Explain. Explaining why auxin causes changing in length. This is where we need to say that auxin causes the cell wall to become more flexible so that cells can elongate, thus causing an increased length. And then a suggest, what was the suggest again? Oh, suggest how results might be differed if lengths of root had been used. So root cells, auxin has the opposite effect of root cells. So in roots, the adding auxin will probably not cause an elongation. And it might actually, I don't think it would cause the cells to shrink, but it would, yeah, it doesn't cause the cells to shrink, it just inhibits elongation. So in roots there would be no elongation, and maybe you could maybe say, and maybe a reduction in length. I don't think that would actually happen, but it might be on the mark scheme, a reduction in length. Let's check the marks. Okay, right. So IAA stimulates cell elongation. That's a mark. In roots, growth is inhibited, or elongation is inhibited. Now this is the horrible thing. Increase in IAA concentration causes a greater mean change in length. Why you have to have mean, I have no idea. Well, it's just you just need to write, when you're writing about the results in AQA, they are really picky on basically quoting the table headings as they are. And if you abbreviate them in any way or shorten them in any way, you tend to lose marks, which is annoying. So the greater the IAA, to be safe, you would write something like this: the greater the IAA concentration added to the Petri dish, the greater the mean change in length of the shoot. Okay, quote this stuff. Okay, takeaway point for AQA: got to quote your table headings and axes, and don't abbreviate or shorten, because they'll take marks away. Let's keep going. 3.5. Coffee. The student produced the different concentrations of IAA using a stock, what does it say, 1 gram per dm cubed or litre solution of IAA. 1 gram per dm cubed is equal to a part per 1,000. Okay? Okay. And distilled water. Complete table 2 with the volumes of stock IAA solution and distilled water required to produce 40 centimeters cubed of 10 parts per million IAA solution. Okay, so the first thing we have to do is kind of work out what kind of dilution this is. So we're going from 1 part per 1,000 to 10 parts per million. So 10 over a million, 10 parts per million. So what 1 part per 1,000 is equal to 1,000 parts per million, because there are, there's 1,000 times 1,000 is a million. So what we're doing is we're going from 1,000 parts per million to 10 parts per million. So we're diluting that 100 times, okay? Dividing it by 100. Okay, so that's the first kind of numeracy thing that you need to do, work out the actual dilution. So 100 times dilution. Concentration of IAA solution parts per million, this is what we want, 10. So the volume of the stock solution, and then the volume of distilled water. Well, we only want 40 centimeters cubed total, so we need 1/100th of 40 for this, and then 99/100ths of 40 for this. So 40 divided by 100 is going to be 0.4. And then the rest is going to be made up with distilled water, so that's 9.6. So hang on a minute. We want to make 40 cm cubed, so that's 39.6. There we go. Right, check the mark scheme. Yeah, 0.4 and 39.6. Question 4. A scientist investigated the effect of a decrease in pH, okay, so that's more acidic, on muscle contraction. The scientist did the investigation with 4 different preparations of isolated muscle tissue. Okay, what have we got? Mouse muscle fibers at a typical pH of mouse muscle tissue. Okay, control 1. Normal mouse muscle fibers. And then mouse muscle fibers at 0.5 pH units below. Okay, so this is like, I'm just going to call this acid mouse. And then C, rabbit muscle fibers, typical. All right, so that's like normal, that's control 2, normal rabbit. And then we've got, acid rabbit. Acid rabbit. They measured the force of muscle contraction of the muscle fibers at 3 temperatures, 12, 22, and 32. Strange temperatures to use, but that's what they did. 12, 22, and 32 degrees C. Here are the results. Figure 3 shows the results the scientists obtained for B and D compared with the appropriate control. What does that mean? Ah, I see. So if we look at this axis here, force of muscle contraction is given as the percentage of the control. So like, for example, the acid mouse starts out, mouse muscle fibers here, B, starts out at, what's that, 82? 4, 6, 8, 10, yeah. So that's 82. 82% of the normal muscle fiber when it's not acidified, okay, at 12 degrees C. And then it goes, just to show you this here, this would go to 90% of normal at 22, and then weirdly 130, wait a second, 101, 104, no, 105, 105. Yeah. Okay, let's look at the question. So, but this is weird because, like, it starts off contracting with less force, but then when you get above these temperatures, there and there, when you get above those temperatures, or certainly when you get to 32, the force is actually higher at high temperatures. Weird. Okay. Ooh, okay. A student looked at the results and concluded that a decrease in pH does cause a decrease in the force of muscle contraction. Okay, well, this is a classic evaluate question. Let me just look at this. All right, they concluded that a decrease in pH, I'm getting better at these, I'm practicing these on AQA, and I'm getting better, I'm spotting clues. Here's some clues. First of all, we need to look at the results, okay, and say, does this confirm or deny? Okay, so the results. There's going to be some positive things from the results, and there's going to be some kind of negative things from the results, like a positive for the evaluate and a negative for the evaluate. And then there's this, cause, okay, causality. Okay, we can't really prove. We're looking at correlation. We have no statistical tests. So let's break this down. So let's first look at the results and try and find some positive and some negative. Okay, so the big thing for positive, things for and things against this conclusion is we need to look at regions of the graph. And the key points are where we cross this 100% line. So we need to identify where these kind of trends cross over this line. So first of all, let's look at the mouse. For the mouse, it's there. So if we drop this down to there, that's 21, 2, 3, 4, 5, 6, 26.5. So 26.5 degrees is the key point for the mouse. Below 26.5 degrees, the force is lower, and above 26.5 degrees, the force is higher than normal. What about the... Oh, that's... hang on a minute. That is not the mouse, that's the rabbit. Oops. That's the rabbit. The mouse is this one. So this figure here, let me actually just remove my mark there because I can't quite tell. That looks to be 29, I'd say. Okay, so for the mouse, greater than 29 degrees C gives a greater force of contraction. So there's two more points I would think from that. So we, quoting those numbers, saying that below these values, the force is lower, and above these values, the force is higher. Okay, so the data, a mark for and a mark against. Let's scroll down. So sort of below 26.5/29, force is lower. And then above these values, force is higher. Now this thing, does it cause a decrease in the force of muscle contraction? there is no statistics here. So in order to prove that there is causality and the results are significant, we need to do a statistical test. So that you'd say there's no statistical test, therefore we can't prove that there is a significant, we cannot prove that the results are significant. Okay? And the other thing is, which I just recently spotted, is that this student is concluding things about muscle in general, but we only looked at two types of muscle: rabbit and mouse. So we don't know if this conclusion applies to human muscle or other types of muscle. And final thing I might throw in as well, just as a guess, is that I kind of don't like the temperatures that this investigation is done at, because the maximum temperature is 32, but mouse and rabbit muscle fiber in real life would be pretty close to 37 degrees C, because that's body temperature for mice and rabbits and humans and mammals. So why are we doing this at 32? It seems strange. Let's have a look at the mark scheme and see what points are available. Whoa, okay, there's 7 different things that we could say. Right, what have we got? Okay, so so 1 mark for talking about where the graph is below that 100, so below 29 or below 26.5 degrees C for mouse and rabbit. 1 mark for saying that it's higher above those values, higher above 29, higher above 26.5. A mark for saying that it's only mouse and rabbit used, no other species is used. And that kind of last guess that I threw in there, it did come up. So body temperature of mouse or rabbit would probably be higher, probably be 37. Oh, these were some good ideas. I didn't think about these, but these could have, that makes sense. Okay, so let me just point out what the student said. The student said that a decrease in pH. We only decreased the pH by 0.5 degree. So how do we know if it would carry on 1 pH unit below or 2 pH units below? So we only use 1 pH, not multiple pHs. Should have used more pH values. And we use isolated muscle tissue in this investigation. So that means it wasn't in vivo, it was in a petri dish or something like that, I presume. And that final mark for the stats test, it was there. We have to make sure that we use this word significant. So there was no statistical test to see if the results or the difference is significant. Okay, let's keep going. Oops. Another group of scientists suggested that a decrease in the force of muscle contraction, is caused by an increase in the concentration of inorganic phosphate. Okay? Their hypothesis is that an increase in the concentration of PI prevents the release of calcium ions within the muscle tissues. It's an interesting hypothesis. Okay, so remember, calcium ions are released from sarcoplasmic reticulum. Okay, I've got that animation ready for you now. So how does calcium normally work to initiate muscle contraction? So the thing is that the actin fiber is normally covered by troponin and tropomyosin. So in this animation here, tropomyosin covers the myosin head binding sites on the actin, and troponin, these little blue proteins here, they're not actually blue, just colored blue in this animation, is sitting on top of tropomyosin. So when calcium ions are released from the sarcoplasmic reticulum, they can bind to troponin, and they cause a subtle conformational change in shape so that the tropomyosin moves and reveals the binding sites on the actin filament. And it's only now that the myosin head can bind to the actin and cause the power stroke, moving along the actin filament. Okay, I just might as well play this out for you. So we get it moving along. ATP binds, actually releases the myosin head, cocks it back into the ready position, binds again, and it drags, the myosin head kind of drags itself along the actin filament, doing this power stroke cycle, or you could call it the cross bridge cycle, again and again and again, until it finally stops when calcium ions are actually actively transported back into that sarcoplasmic reticulum. Okay, so let's check the mark scheme. So, troponin and tropomyosin moved from the binding site, or is not removed from the binding site, or the shape of the tropomyosin not changed. So in AQA, you don't actually need to know about troponin, even though it's troponin that actually binds the calcium. It's tropomyosin that changes shape. Okay, so you do need to mention tropomyosin. If you mention troponin and tropomyosin, no problem, but tropomyosin is the thing that you need to mention. So less tropomyosin is moved away from the binding site, or the shape of the tropomyosin is not changed. So the myosin head cannot bind. This is, I'm not sure if I would have said this, actinomyosin bridges. I would have said cross bridges. I'm not sure I would have got the mark. Was it a 3 marker? Yes, it was a 3 marker. So this is some language you need to learn. I need to learn. Actinomyosin bridges. Fewer or no actinomyosin bridges formed. Actinomyosin bridges. So the myosin head does not move or pull on the actin filament, therefore not generating a force. Myosin does not pull the actin, or there is less ATPase activity. Actinomyosin bridges, something I need to remember. Actinomyosin bridges. All right, moving on. .3. In muscles, pyruvate is converted to lactate during prolonged exercise. Explain why converting pyruvate to lactate allows the continued production of ATP by anaerobic respiration. Okay, so when we convert pyruvate into lactate, we are reducing pyruvate into lactate. So we're adding hydrogen atoms to pyruvate to produce lactate. Let me just draw this. Pyruvate. We basically need to remember this. And in so doing, we use up reduced NAD. Reduced NAD, and we convert it into oxidized NAD, or just NAD. Okay, we're getting rid of reduced NAD and regenerating oxidized NAD. That is crucial, because that oxidized NAD is needed for glycolysis. So glycolysis, which does produce ATP, needs NAD so that it can, it's in the final stages of glycolysis when triose phosphate is converted into pyruvate. That's it. Okay, let's look at the mark scheme. So it regenerates or produces NAD so the glycolysis can continue, except description of glycolysis, e.g. glucose to pyruvate, for oxidizing, converting triose phosphate to pyruvate. Okay. Easy mistake to make, just so I'm going to highlight this over here. Reject NADP, okay, because NADP is photosynthesis, and also reject any reference to FAD. FAD is not involved in glycolysis. It is a coenzyme. It is involved in the Krebs cycle, but not in glycolysis. Okay, on to question 5. Describe the role of glucagon in gluconeogenesis. So this word, long word, gluconeogenesis. Glucose, neo meaning new, genesis meaning making new glucose. So this occurs in the cells of the liver, and it is in response. So let me read this question. Do not include in your answer details of the second messenger model of glucagon action. So we don't need to talk about cyclic AMP. We just need to talk more specifically about the end result, gluconeogenesis. So I would probably say, just to cover my bases, glucagon is released from alpha cells in the pancreas. It binds to receptors on cells in the liver. It activates enzymes. These enzymes convert fatty acids and glycerol and amino acids into glucose. I think that's about it. That should do it. Let's check, see that that's right. Okay, it's mainly the marks available are just for this kind of enzymes and the substrates that are converted into the product, which is glucose. So 1 mark for activating enzymes, and the second mark for any of these 3 things into glucose. You could have talked about glycerol or amino acids or fatty acids are converted into glucose. (5.2). The gene that codes for glucagon is 9.531 kilobases in length. So that's 9,531 bases in length. The DNA helix makes 1 complete turn every 10 base pairs. Every complete turn is 3.4 nanometers in length. Okay, cool. And then we just need to work out the length of this DNA. So it's 9,531 divided by 10 times 3.4 nanometers, and that's going to give us a length in nanometers, and then it wants us to convert it into micrometers just for fun. Let's first do the nanometers one. 9,531 divided by 10 times 3.4 equals, so it's 3,240.54 nanometers. Okay, and then to convert that into micrometers, we need to divide it by 1,000, because there are 1,000 nanometers in a micrometer. So that's 3.24 micrometers. Now, how does the question want it? 3 significant figures. Okay, so that is correct. Always check the level of significant figures for calculations. It's very easy to lose marks. 3.24. Let's see if that's right. Cool. Yeah. And if you had failed to spot that significant figure bit, you might have got 1 mark. All right, moving on to 5.3. Metformin is a drug commonly used to treat type 2 diabetes. Metformin's ability to lower the blood glucose concentration involves a number of mechanisms, including increasing a cell's sensitivity to insulin, inhibiting adenylate cyclase. Explain how increasing a cell's sensitivity to insulin will lower the blood glucose concentration. So, if a cell is more sensitive to insulin, then a certain amount of insulin will cause more glucose transport proteins to be incorporated into the cell membrane, making the cell more permeable, resulting in more glucose entering cells and resulting in more glucose being stored as glycogen. I think we're just looking for repeatedly using the word more, really, and then kind of following through the steps. More of this, so more of this, so more of this, so more of this. Okay, let's look at the mark scheme. Okay, so definitely get a mark for point 2. Stimulates uptake of glucose by channel or transport proteins. But I'm a bit perplexed that most of the marks are just for describing the normal mechanism of insulin action. So insulin binds to receptors which then causes the addition of channel or transport proteins into the membrane, and it also activates enzymes which convert glucose to glycogen. So this is really just how insulin works. I'm slightly intrigued that this question just wanted you to say how insulin works and nothing about sensitivity to insulin. Okay, so I suppose a good strategy for this type of question is to sort of describe the normal mechanism and then suggest how it would be increased. So when insulin binds to receptors on the cell surface, this triggers the incorporation of transport proteins into the cell membrane and the activation of enzymes that convert glucose to glycogen. If the cell's sensitivity is increased, there will be more transport proteins and more activation of enzymes, resulting in a greater intake of glucose. That would be a good answer. Question 5.4. Explain how inhibiting adenylate cyclase may help to lower the blood glucose concentration. Okay, so I wanted to put in a picture over here, which I've done, to kind of take you through this. So this is beyond A level, this is kind of a university level diagram. You don't need to know so much about it, but there are some things that are on the A level spec. So either glucagon or adrenaline actually work in this same way. When they bind to receptors, they trigger the conversion of ATP into cyclic AMP, which happens inside the cell. And this enzyme here, AC, this is adenylate cyclase. So that's the protein that causes the production of this second messenger, cyclic AMP. It then activates a kinase enzyme. It's actually called protein kinase A, but you can just call it a kinase. And it's this kinase that then kind of initiates a cascade of other reactions that eventually cause the activation of this, what's it called? Glycogen phosphorylase? Actually, I can't remember what the name of it is. It causes the activation of an enzyme that breaks down glycogen into glucose. Is it glycogen phosphorylase? I forget exactly the name of the enzyme. But it's taking glycogen and converting it into glucose. That is going to increase glucose concentration of the bloodstream. So, inhibiting adenylate cyclase may help to lower the blood glucose concentration because with inhibited adenylate cyclase will produce less cyclic AMP. With less cyclic AMP, we get less kinase activation, or fewer kinase enzymes are activated, and this will result in less glycogen breakdown into glucose. And this word, glycogen is glucose, we can describe it with a word, which is glycogenolysis, splitting down of glycogen. Okay, let's check the mark scheme. Okay, so less ATP into cyclic AMP, less kinase is activated, and less glycogen converted into glucose. This answer is more kind of what I was expecting for 5.3, to be honest, is less, and less. I was expecting for 5.3 more, and more, but it was a bit strange, that one. Okay. Question 6. Ooh. A pedigree analysis diagram. These ones are fun. Right, okay, in fruit flies, a gene for body colour has a dominant allele for grey body, big G, and a recessive allele for black body, little g. Okay? Big G is grey, small g is black. A gene for eye colour has a dominant allele for red eyes and a recessive allele for white eyes. Red eyes and white eyes, and is located on the X chromosome, so this is sex linkage. Alright, so we're going to have to look at two different alleles and consider sex linkage. Ouch. So we've got big G, little g, that's normal. Then we've got big R, little r, that's sex linked. Okay, here shows the phenotypes of fruit flies over 4 generations, and we've got a key over here. and then we just the key, and then we just got the body color is just written on. So the shading is the red, white, and the body color is just written on. So be careful not to like just confuse that. Okay, let's look at the question. Give the full genotype of the fly numbered 6. This one. Okay, so what do we know about this fly? It is grey body, and it is female with red eyes. Okay, so the fact that it is female with red eyes, this is problematic because it has the dominant characteristic. It has the dominant characteristic grey body, and it has the dominant characteristic red eyes. But we don't know, is it homozygous or heterozygous? That is what makes this tricky. So is there a way that we can figure out whether it's homozygous or heterozygous? The easiest way is probably to look at its offspring. So let's look at the cross that it can produce. Now, it does produce both grey and black body offspring. This one here has a black body, so this is like GG. Now, if this was big G, then there would not be able to be any black body offspring. So this one has to be heterozygous. So it's got to be, we can already write that in, big G, little g. And we know that it is female with red eyes, so it has to have an X with a big R. Now, what about the last X? Does it have a little r or not? Does it? I think it must do, because to produce this fly here, female with white eyes, it has to pass on an X with a little r, and so does this one. This would also have to pass on an X with a little r. So it must be heterozygous at both alleles, at both loci. Gosh, that's for 1 mark. These questions are really hard. I guess key takeaway strategy, if you don't know if it's homozygous or heterozygous, look at the offspring and figure it out. Okay, that will be the best clue. Give one piece of evidence from figure 4 to show that the allele for grey body colour is dominant. Okay, I think I've got one. So if we look at this crossover here, what I was looking for was, have we got a place where we've got 2 grey body parents having a black body baby fly? And yes, we do. So over here, we've got 2 grey body parents, 3 and 4, producing a black body baby fly. Okay, so this must mean that both 3 and 4 are heterozygous for grey body, and that means if they are heterozygous and grey bodied, it means that the grey body is dominant. Right, let's check the mark scheme for both those, 6.1 and 6.2. Okay, so yeah, this was just heterozygous for both loci. So yeah, 3 and you could just say 3 and 4 produce a black fly. Grey parents produce a black fly. What's next? Explain one piece of evidence to show that the gene for body color is not on the X chromosome, so it's not sex linked. All right, this is a very tricky question. I'm trying to get my head around it. So I think I'm going to approach it by thinking, what would happen if body color was on the X chromosome? If body color was on the X chromosome, if it was, it's not, but what if it was? Then we'd have something like this, X big G, X little g, and we'd do like a cross, and maybe we'd have something like this. I don't know, what would this look like? This would look like... So that would mean that a black-bodied male would only, no, would only, no, but this was, maybe we want to think about a, okay, actually, yeah, let's think about this. If we had a black-bodied female, yeah, there we go, X, like that, and a grey-bodied male, then this would be correct, and this would be correct, but that would be, okay, so if we had a black-bodied female, that's what we're looking for, okay, black-bodied female and a grey-bodied male would only be able to produce a black-bodied male. Does that appear up here? So I'm looking for a grey-bodied male and a black-bodied female. Aha! Grey-bodied, oh no, that's the grey-bodied. I want a grey-bodied male. Grey-bodied male. Here we go, grey-bodied male and a black-bodied female. So the grey-bodied male and the black-bodied female, we would only, if it was sex-linked, we would only be able to produce we'd only be able to produce black-bodied males. Is that what we've done? No. Here's the grey-bodied male. Oh my gosh, that was so hard. All right, so this is the pairing that I'm looking at. Okay, grey-bodied male and black-bodied female. If it was sex-linked, this is not possible. There. Okay, that would be impossible. That is a very hard question to articulate, and it's not something that you can just remember, I don't think. So explain the evidence that it's not on the X chromosome. Strategy. Think of what it would be like if it was on the X chromosome. What would be the musts, what would be the rules, and then try and find an exception to that rule. There's probably a few. Which one did I actually talk about? I said fly 1 and 2 produce 5 is what I was talking about. Is that even on here? Yeah, okay, so I got this. Fly 2 and 1 produce 5. So a black female produces a grey male. So fly, I didn't explain why. So because fly 5 would inherit the recessive allele from 2. Right, this is a super hard question to kind of articulate. So if that comes up, just do your best. Let's move on. It's making my head hurt. 6.4. A heterozygous grey-bodied white-eyed female fly was crossed with a black-bodied red-eyed male fly. Complete the genetic diagram below to show all the possible genetic genotypes and the ratio of phenotypes expected in the offspring from this cross. Okay. Heterozygous grey-bodied white-eyed female fly. Okay, so let's first of all work that out. So grey-bodied, heterozygous it says, so that's big G, little g. White-eyed female fly, so that's X with a little r because it's white. And it is recessive, that, so it's got to be XR, XR, like little r, was crossed with a black-bodied, red-eyed male fly. black-bodied has got to be little g, and it's red-eyed and it's male. So it's got a big X with an R, and because it's male, that other one is a Y. Okay, this is nicer. So now we think about what gametes can be produced, and actually it's not so complex because there's only 2 different gametes that can be produced from each fly. This fly can either give you a gamete with a big G and an XR, or it can give you one with a little g and an XR. Okay, so those are my gametes. It's always a good idea to draw a circle around your gametes. It's just kind of genetic convention. Over here, we've got a little g and an X big R, or we could do a little g and a Y. Okay, I like a sort of Punnett square method, so I'm just going to draw my Punnett square like this. Basically like flip this gamete, put it there, flip that gamete, put it there. So now what are my possible combinations? Let's zoom in a little bit. My possible combinations here would be big G, little, oh, what am I doing? I'm looking at this one, little g, and then on the X chromosome, we've got big R, little r. That. Split this into 4. This one here, I've got little g. I've got an X with a little r, and I've got a Y. And then over here, I'm going to have big G, little g, and I'm going to have X, big R, X, little r. And over here, I'm going to have big G, little g, and I'm going to have an X with a little r, and I'm going to have a Y. So write those genotypes down here. What are the phenotypes? Let's look at them 1, 2, 3, 4 in that order. So 1, this phenotype is going to be black-bodied, red-eyed female. Black-bodied, red-eyed female is number 1. Number 2 is going to be a black-bodied, white-eye male. A black-bodied, white-eye male. Number 3 is going to be grey-bodied, red-eyed female. And number 4 is going to be grey-bodied, white-eyed male. And the ratio of phenotypes for this would be 1 to 1 to 1 to 1. Okay, there's the mark scheme. So you get marks for actually working out the parents, I think. Yeah, that's the marks for working out the parents. 1 mark for that. Then you get marks for the genotypes of the offspring, and then for the 3rd mark you need both the description of the phenotype and the ratio. You need all of this for the 3rd mark. Okay, moving on. Okay, 6.5. This says a population of fruit flies contained 64% grey-bodied flies. Use Hardy-Weinberg equation to calculate the percentage of flies heterozygous for gene G. Okay, so first of all, remember Hardy-Weinberg, 2 things you need to remember. P plus Q equals 1, and then we've got P squared plus 2PQ plus Q squared equals 1. So first thing you need to do is figure out what you've been given. Now, it's a little bit tricky because 64% grey-bodied flies, if you're a grey-bodied fly, you might be homozygous, in which case you'd be big G, or you might be heterozygous, in which case you'd be big G, little g. So this 64% is not your P squared. It's actually this. It's actually like your P squared plus your 2PQ, because this is the P squared is the big G, and the 2PQ is the big G, little g. So actually, what you want to do is 100% minus 64% is equal to your Q squared. Okay, so what we do know is who is going to be little g, okay? The little g, black flies, we know the percentage of black flies is 100 minus 64, which is going to equal 36%. equals Q squared, okay, which is the black flies. So therefore, Q is equal to the square root of 0.36, which is actually 0.6, yeah, that's right, which is equal to 0.6. And then we want the percentage of flies heterozygous for gene G, which is 2 times P times 2, 2 times P times Q, which is equal to 2 times Q is 0.6, so P is 1 minus 0.6, which is 0.4 times 0.6. Okay, so this is 2, this is P, this is Q. 2 times 0.4 times 0.6, which is going to be 0.48, or 48%. There we go. Correct answer, 48%. Ta-da! Now, if you didn't get that right, you might have got 1 mark, but shows an understanding that 2PQ is the heterozygous, or doing something that shows your what does this even mean? Incorrect answer, but shows understanding that 1 minus P squared plus 2Q squared equals, okay, yeah, fine. Yeah, so some understanding. Okay. Let's keep going. On to question 7. Okay. Question 7. In photosynthesis, which chemicals are needed, I think that's important, needed for the light-dependent reaction? Not produced, but needed. So in the light-dependent reaction, we're phosphorylating ADP, so we're definitely going to need ADP and PI. It's not ATP, it's not ATP. So it's that. What else? We're reducing NADP. So we don't need reduced NADP. We want oxidized NADP. Okay, oxidized NADP, ADPI, and yes, water because we're doing photolysis. So yes, it's this one. That one. Okay, keep going. Describe what happens during photoionization in the light-dependent reaction. 2 marks. I've just dropped in a little image over here to help you kind of visualize that. So we're having photons, I would say, photons of light are absorbed by chlorophyll molecules, causing electrons to be excited within the chlorophyll molecules. And when they are excited enough, they leave the chlorophyll molecule and are passed on to the electron transport chain. Okay, this picture here kind of shows that electrons can also be passed to DC-PIP if you're investigating this process, but if you're not, then they would just be passed down the electron transport chain, going this way, going to the next photosystem. Okay, so that's photoionization. Okay, let's check the mark scheme for the exact language that you need. So we need chlorophyll absorbing light. Don't have to say photons actually, but just chlorophyll absorbing light or photons of light. Light excites electrons in the chlorophyll molecule. This is all 1 mark point actually. And the electrons are lost, or passed. This would have been accepted, except electrons go to the electron transport chain. And, or you could have said chlorophyll becomes positively charged. I'm surprised that chlorophyll becomes oxidized wasn't accepted as well. I would probably just always say both. The mark schemes in AQA, they do vary quite a bit. Sometimes oxidized might be credited, sometimes not. So I'd probably say electrons are lost as chlorophyll becomes oxidized and positively charged. You just want to like put all of this in a sentence because you're going to get the mark scheme. You're going to get the marks somehow. Okay. On to this. Okay, chromatography. A student obtained a solution of pigments from the leaves of a plant. Then the student used paper chromatography to separate the pigments. Here's the chromatogram produced. Explain why the student marked the origin using a pencil rather than using ink. So ink is soluble in the chromatography solvent, and the pigments in the ink would move up with the solvents, meaning that you couldn't see the position of the chloroplast pigments, is what I would say. So quite a lot there for 1 mark, but I'm just making sure that I'm going to get it. Did I miss one? Ink and leaf pigments would mix. I think what I said would get the mark. Have I missed a question here? 7.3. I think that would get the mark. What do the other things actually mean? So I guess the origin line would, if it was origin line as in pen, it would move, I guess is what they're saying. The whole origin line would move up if it was in pen, I suppose. Right, let's just keep going. 7.4, describe the method the student used to separate the pigments after the solution of pigments had been applied to the origin. Okay, so you would place the chromatography paper in chromatography solvent in a tank. You're making sure that the chromatography solvent is below the line of the origin. You would wait for the chromatography solvent to move up the chromatography paper, and then you would remove the paper from the solvent before the solvent front reaches the end of the chromatography paper. Let's have a look. Yeah, those are the 2 marks kind of needed. When you put it in the solvent, it's got to be below the start line, the origin line, and then you've got to remove it before the solvent reaches the top end. It does say interesting over here, reject water or any named aqueous solution. That is not the solvent. The solvent is normally something else, something kind of nasty. Normally like propanone and some other solvents kind of mixed together, but something like that, so not water-based. Sorry, 7.4. No, 7.5. Calculating the RF values of the pigments can help to identify each pigment. An RF value compares the distance to pigment, blah, 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 blah. Okay, yeah, cool. The distance each pigment has moved is measured from the middle of each spot. Something worth remembering, they don't always tell you that. Pigment A has an RF value of 0.95. Use figure 5 to calculate the RF value of pigment C. Okay, so this is quite a lot of work for 1 mark. I haven't got a ruler on my kind of digital version here, but I'll show you what you would do. So A has 0.95. Okay, so first of all, you measure the distance to the middle of pigment A from the origin line. Let's call that length A, okay? So that's the distance that the LA length A has moved. So the RF value is going to be equal to the length of A. I'm going to write over solvent front, okay? And now we know that the A has a RF value of, was it 0.95? Yes, it was 0.95. We know that is equal to length A, which you've now measured, divided by the solvent front. So then you can rearrange to get solvent front. So therefore solvent front is equal to 0.95 divided by length A. That will give you some value for the solvent front. Let's, I don't know, whatever it is, 6 centimeters, let's say. And then you can use that value to work out the RF value for pigment C. So again, for pigment C, you're going to have to actually measure it to the center of the spot. We'll call that length C. So now you do RF value equals length C divided by the solvent front. Remember, you've already got that solvent front there. Put it in there. And there you go. That's how you do it. Mark scheme says that you should get somewhere between 0.58 to 0.62. Okay, so measuring accurately is important. Typically speaking, in A-level biology papers, any measurement that you have to do with your ruler is only given a millimeter tolerance maximum. So it is really important that you take your time to try and measure it accurately. Okay. The pigments in leaves are different colours. Suggest and explain an advantage of having different coloured pigments in leaves. You just need to remember, key phrase is that they absorb a wider range of wavelengths. Each of these pigments absorbs a slightly different wavelength. Quite often this is like chlorophyll B down here, carotenoids up here. They absorb different types of light. That's the answer, really. So different, more wavelengths, except a wider, larger range of wavelengths. Yeah. I'm interested that you have to say photosynthesis. Yeah. There we go. I'll edit that bit out.
Sorry. Didn't want to be
Fuel.
rude.
Huh?
Didn't want to be rude and not offer you.
Oh, right. I'm very kind. Oh, good. Good strawberries as well. It's peak strawberry season.
Yes.
I'm interested that you have to say photosynthesis. So actually, it doesn't. It's not underlined. I wonder if you get the mark for saying can absorb a wider range of wavelengths and just left it there. Would you get the mark? I don't really know. To be safe, try and remember, absorb a wider range of wavelengths for photosynthesis. Okay, on to question 8. What is a DNA probe? So a DNA probe is a single-stranded piece of DNA that can bind to a target sequence through complementary base pairings. I think that's what I would say. So this can bind over here, and I would add the DNA probe is normally labeled in some way, such as with fluorescence or radioactivity. So this could be radioactive phosphorus. Can't remember which one that is. Phosphorus 35, but radioactive phosphorus, or it could be a fluorescent molecule attached. Let's see if that gets the credit. A single strand of DNA. I wouldn't have. Did I say that? I said it would bind via complementary base pairing. I think that would be credited because I said base pairing, but you have to say the bases are complementary. Not that the DNA is complementary, but the bases on the DNA probe are complementary to the target sequence. In this question, it didn't require you to know the different types of labeling on the probe, but it is normally fluorescence or radioactivity. Okay, DNA probes are used to detect specific bases of DNA. And here's the process. A DNA sample is broken down into smaller fragments. Fragments are separated via electrophoresis, are transferred onto a nylon membrane. The DNA fragments are treated to form single strands, and then the DNA probes are added. And these probes allow specific base sequences of DNA to be detected. Describe how the DNA is broken down into smaller fragments. That's one that's tricky. I'm not sure if I remember it. I'm going to say restriction enzyme digest. Yeah, restriction enzymes. Is there another way? I don't think so. So restriction enzymes. And then it's a 2 mark, so I want to add more content. So I'd say restriction enzymes cut at specific sequences, specific palindromic sequences, maybe, just to be sure, or specific recognition sites, you could say. Okay, so it is restriction enzymes. Restriction enzymes cuts at specific base sequence. You could say that it breaks phosphodiester bonds at recognition sites. (8.3). The DNA on the nylon membrane is treated to form single strands. Well, this is so that you want to separate the the double-stranded DNA to expose the bases on the inside so that the probe can then come in and bind on top of that. Okay? The language that we need. Okay, so the probe binds attaches or anneals. Yeah, maybe worth, if you haven't seen that word anneal, just you might not need to use it because binding will do, but you could be given that word. Annealing means when complementary bases bind to each other. Annealing. Oops. A scientist used DNA probes and electrophoresis to screen 4 volunteers for 5 different viral DNA fragments. Okay. Figure 7 shows the results the scientists obtained. So we've got the 4 volunteers there. The lanes numbered 2 to 5 represent the 4 volunteers. What's lane 1? Doesn't say. Oh, lane 1 of figure 7 enabled the size of the different viral fragments to be determined. So, okay, yeah, so often in gel electrophoresis you have like this kind of reference column where you've got known sizes of bases. So like it might be that you know that this one here is 100 kilobases long. This one is 80 kilobases long, kilobase pairs, and then 60, 40, 20, 10, maybe this is 5, maybe this is 2, or something like that. Remember that the smaller fragments of DNA move through the gel more quickly, so it would be in that order. So I kind of just explained it. Let's see what language you need to say or use. Sorry. So it has DNA fragments of known sizes or lengths. Yeah, and you need to say that you would compare the position of the probed fragments with the known fragments. So, for example, going with what I just said, if this one here, if you go with the scale that I kind of made up, that would be a 20 kilobase pair fragment, pretty much, more or less. In fact, they all line up exactly, don't they? Or if it was like halfway, if it was like there, you might say that's about 15, for example, between 10 and 20. Oops, what's the next question? Oh, it tells us the lengths right there. Okay, so here are the lengths. I'm going to rub out my lengths and write on the lengths that it actually gives us. What have we got? 6 to... Oh, what on earth? It's really strange. Strange scale. Okay, so we need to put them in order. 605. This is weird. So this is 600. This is 535. What's the next one? Then it goes 500, and then 300. This is 500. This is 300. What's next? That's 250. Oh no, I've done it wrong here. I now understand what I've done, because it's these bits. These are the bits. Okay, so we've got to do, this is the 600. This is the 535. This is the 500. What? This is the 300 here, and this is the 250. There we go. Let's see what that is. And then it says which volunteers had at least one of the viral DNA fragments with 250 or 535. Okay, so which ones had it? This is such a strange question. So that's a 250, that's a 250, that's a 535. So which of the volunteers, 3, 4, and 5? 3, 4, and 5 had at least one of those. What an odd question. Is that right? 3, 4, 5? Yeah, it is. 3, 4, 5 with these numbers in any sequence. All 3 numbers required, because it says at least one. Okay. On we go. The sundew is a small flowering plant growing in wet habitats such as bogs and marshes. The soil in, here's a sundew, by the way. The soil in bogs and marshes is acidic and has a low concentration of some nutrients. It's going to be nitrates. The sundew can trap and digest insects. Describe how you could estimate the size of a population of sundews in a small marsh. Okay, so basically, first of all, you would have to look at your marsh, kind of measure its size, measure the area that you want to survey, its length and its width. You'd put an axis down here with a transect line, an axis down here with a transect line. Use a random number generator to pick a point on this axis. If you haven't seen this before, this is what I'm talking about. So let's say this is, I don't know, 80 meters long, this little marsh. Well, you basically just use your random number generator on your calculator. It is there, it says rand. Let me just show you. Where is it? There it is, rand. You get some number like that, random number, 0.99 times that by 80. So that would give you a coordinate on this axis, and it happens to be 79, so right up here. So we go down there. You do the same thing here. Let's say this is like, I don't know, 30 meters long. You do the same thing here, pick a random number, times it by 30, maybe you get a coordinate here. You move to the intersection of these two coordinates, and you place your quadrat. You place your quadrat, and you count the number of sundews. You've got one data point. You do this multiple times, each with two different random numbers giving you different coordinates. You build up a pattern of sampling that is random. And then you work out the average or the mean number of sundews per quadrat. And then if your quadrat is 1 meter squared, let's say, you times the average number of sundews by the total area to get the estimate for the population. Okay, so that's how you would do it. Let's look at how you describe it, kind of using the right language. Use a grid or divide the area into squares or sections. Method of obtaining random coordinates. For example, random number generator is the easiest one to remember. Count the number of sundews in a quadrat. This, I'm not sure if I emphasize this enough. Make sure that your sample is large and calculate the mean or average. I did say calculate the mean, but it has to be a large sample size. Yeah, as a rule of thumb, like at least over 10, probably more. I think there is in ecology, if you study it, there's like you should cover a certain percentage of the area or something, like maybe it's over 5% or over 10% of the area you should sample, something like that, but a large sample. And then, for example, the mean number of plants per quadrat multiplied by the total number of quadrats in the marsh, or something like that, or mean number of plants per meter squared multiplied by the total area in meters squared of the marsh. Just maths, really. I'll zoom out a teeny bit just so you can see all these different accept or do not accept comments. There's quite a lot of them down here. Oh, look here, this is interesting. Just spotted that. So for large sample, if a specific number is given, it must be 10 or more. So that is the AQA definition of large is more than 10. Okay, let's move on. Okay, suggest and explain how digesting insects help the sundew to grow in soil with very low concentrations of some nutrients. I think we're mainly going to be talking about nitrates. So sundew grows in soil lacking in nitrates. Insects' bodies contain nitrates in the proteins. So when the sundew traps the insect and digests it, breaks the proteins down into amino acids. Those amino acids can be further broken down to provide a source of nitrogen, or to provide amino acids for growth. Yeah, I think that's should do it. Let's double check what the mark scheme wants to. Okay, so digestion, breakdown of proteins, provides amino acids. As I just said, amino acids for growth. There are a couple other mark scheme points there that you could say. It is out of 2, so that would get 2 marks, but provides amino acids, or you could say provides nitrogen for the synthesis of DNA, for example. You could also have talked about phosphate, digestion of a phosphate-containing compound like DNA provides phosphate maybe for production of ATP. Yeah. But generally it's nitrogen. Just going back to that, you can get asked about this. It is linked to the nitrogen cycle. I'm surprised. I was expecting a question on this. Wet habitats tend to have more denitrifying bacteria that remove nitrates from the soil, because denitrifying bacteria take nitrates and turn it back into nitrogen gas. And this can only happen in anaerobic environments. So wet, boggy, anaerobic environments tend to have very little, very small amounts of nitrogen. That's why you get these carnivorous plants. We call them carnivorous plants. Even though they don't actually eat the insects, they just get the nitrogen from them. They tend to be in wet habitats. Links to the nitrogen cycle. Okay, right, we're on to the last question of paper 2. The last question of paper 2 is always a comprehension question. Okay, so first of all, let's talk a little bit about what we do in a comprehension question. Zoom in a teeny bit. Okay, comprehension question, obviously you want to read the question carefully, but I advise you to read, highlight, and annotate as you go. It's going to make the next few phases of the question easier, so let's just do that. So what we'll do is you read each paragraph, then I'll come in with my annotations, and then you read the next bit, and so on. So off you go, read the first paragraph. Okay, what bits did you spot? Okay, so it's a rare disease in which the immune system damages the myelin sheath of neurons. So immediately, myelin sheath, for me, brings saltatory conduction to mind. Because if we don't have myelin, we're not going to be able to do that saltatory conduction, which is faster. And if we're going to remove that, we're not going to be able to do that. Numbness, muscular weakness, muscular paralysis, and that's because obviously nerves connect to muscles, so no electrical impulse to muscle. Sometimes neurons of the autonomic nervous system are affected, causing heart rate irregularities, so things like the accelerans nerve plus the vagus nerve that send impulses from the cardiovascular center to the SAN, they aren't working. So CV center to SAN, they aren't working, they're not communicating with the heart, hence irregularities. Let's keep going. Let's read quickly now the next 1-2 little paragraphs there, and I'll come in with some annotations. Have a go, see what you pick out. Okay. All right, let's pick a different color. Okay, Huntington's disease is a disorder caused when a protein called huntingtin damages the brain. It is a dominant mutant allele. I might even just write to the side, like something like this, HH, like that. Big H, little h, has it, okay? Heterozygotes have the condition. The first successful drug trials reduced concentrations of huntingtin in the human brain involved 46 patients. Not a huge amount, it's a few. The patients received the drug for 4 months. Okay, so it's fairly short to mean term. You know, I guess if we really wanted to look at this, you know, we want to know if it works over years, not 4 months. The concentration of huntingtin was reduced in all patients. Well, that's pretty strong evidence. That sounds pretty good. How did they actually give the drug? The drug was injected at the base of the spine into the cerebrospinal fluid bathing the brain and the spinal cord. Okay, this is probably new information to you. You may have heard something about something called the blood-brain barrier. So the blood-brain barrier is actually like, well, a series of membranes and barriers really that prevent what's in the blood plasma from automatically passing into the fluid that bathes the brain. It's like an extra layer of protection. So the reason they're doing this is to get around that to make sure that this drug can actually get into the fluid and doesn't just stay in the blood plasma without reaching the brain. Okay, what about this drug? How does it actually work? The drug contains single-stranded DNA, and they inhibit the messenger RNA. Okay, so this means this little thing here, this is siRNA. You should know about that. Now read the last paragraph. Okay. So let's look at what you should have spotted. Change color again. I'm going to go yellow again. Okay, symptoms of Huntington's disease can start at any time, but they usually develop between 30 and 50 years of age. The likelihood and age when symptoms start are linked to the number of CAG bases sequence repeats in a gene for Huntington's disease. So this is called a triple nucleotide repeat or a CAG repeat. So basically in the gene, it goes like this: CAG, 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 etc., etc. Look it up if you're interested. I think if you have fewer than something like 18 repeats, you do not have Huntington's. And everyone has a bit of repeats in their gene, but if you have more than 18, then that is considered Huntington's, and you will probably develop symptoms at a later stage. If you have like more than 30, it'll come earlier. If you have, I can't remember what the maximum number that's ever been found, but in some poor individual had, I don't know, like 60 repeats, and their Huntington's disease symptoms started in their 20s, for example. However, recent studies have suggested that epigenetics may also affect the age when symptoms start. So epigenetics, we just need to remember what that means. So that's to do with DNA winding around histone proteins, and we need to be thinking about methylation of the DNA. We need to be thinking about acetylation of histone proteins. Okay, so we've done a lot of work already to kind of understand this piece of text, kind of connecting it to knowledge that we have from the spec. Let's go into it. So 10. Damage to the myelin sheath of neurons can cause muscular paralysis. Now it does help by telling us where we need to go, lines 2 to 4. But we've already kind of talked about this, haven't we? Muscular paralysis. We can say that without the myelin sheath, saltatory conduction will not happen, so the nerve impulse or the electrical impulse will travel more slowly, and this means that electrical impulses will reach muscles more slowly or may not even arrive at all, causing no action potential passed on to the muscle and causing muscular paralysis. Right, let's check what they wanted you to say. Okay, so any reference to saltatory conduction, any reference to the nerve impulse being slowed or stopped, and then for that third one, you do need to say neuromuscular junction. So the final passing of the impulse from the neuromuscular junction to the muscle may not occur, so the muscle is paralyzed. You could have said neuromuscular junction or the sarcolemma. The sarcolemma is the membrane around the muscle cell, which is depolarized when the impulse, when we do that chemical signal, that acetylcholine across the neuromuscular junction. You could have said action potential as opposed to impulse. You could have said electrical impulse. But key point here, what not to say. Reject first mark. If you talk about messages or signals, it's too vague. Okay, so let's get that out of your vocabulary. No messages, no signals, electrical impulses or action potentials. All right, back to the questions. What's the next one? 10.2. Heart rate irregularities. Explain why. Again, it tells us lines 4 to 5. We've already done the thinking, so now we just need to describe it. The thinking was up here. We're not sending messages from the cardiovascular center to the SAN down the autonomic system. The accelerans nerve typically speeds up the heart rate. The vagus nerve slows down the heart rate in response to maybe signals that have been detected by chemoreceptors and baroreceptors. I'd kind of be throwing all my knowledge in to this question just to make sure I pick up the marks. Okay. I wonder if I would have got this if I'd written it. So sympathetic and parasympathetic, you need to say slower or fewer impulses or fewer action potentials along the sympathetic and parasympathetic neurons. Interesting. It says reject no impulses, which is interesting. That's really weird. There's a bit of an inconsistency in the mark scheme, because over here, impulses stopped is accepted, but over here, no impulses is rejected. I wonder why that is. Slightly inconsistent. So slower or fewer impulses along the sympathetic and parasympathetic neurons from the cardiac center to the SAN, or from the medulla to the SAN. Now I'm wondering, my brain is still mainly wired for OCR, and in OCR we call this the cardiovascular center. In AQA, I think you'd probably be credited for it, but cardiac center in AQA, cardiac center, I've got to remember that. Cardiovascular center is what it's called in OCR. It's the same thing, of course, but the language they want you to use is very specific. Okay, what's next? Done that one. The first successful drug trials to reduce concentration of huntingtin in the brain used single-strand DNA molecules. This is basically explaining siRNA. So siRNA, what happens? Here's the siRNA. It binds to, is complementary to the messenger RNA, forming some double-stranded RNA, because it binds via complementary base pairing, and that double-stranded RNA is then kind of recognized as foreign and broken down, and translation of the protein does not happen. So, the fact that the DNA is complementary, the fact that it binds to the messenger RNA, and it prevents translation. 10.4. Scientists from the first successful drug reported the drug is not a cure for Huntington's. So just 2 reasons why the drug should not be considered a cure. That's interesting. It says lines 9 to 11. Do not include repeats of the drug trial in your answer. Lines 9 to 11. Okay, so it's just asking you to focus on this particular box here. All right, so it says do not talk about repeats. Okay, well, that's great. So first of all, only 46 patients. Second of all, only 4 months. So it's a fairly short-term trial, and it's only done on 46 patients. It does say suggest 2 things, so I would probably go with those 2 things. But I'm also thinking that the drug, this is a genetic. I don't know if the mark. This is definitely true, but I don't know if it's on the mark scheme. This is a genetic condition. The only way of curing a genetic condition is through gene therapy, and this drug would have to be taken, every week, every month, for life, to keep bringing down the production of that huntingtin protein. You would never stop taking it. It's not a take it once and then done kind of thing. Okay. So only 46 or small sample size, correct. Only 4 months, correct. Yeah, in fact, everything we just said is almost on there. So the huntingtin protein is okay. So the protein, that's interesting actually. Yeah, it did say the protein was reduced, not stopped. So that's an interesting thing you could have picked up on. This is what I was talking about. The allele gene mutation of Huntington's is still present. So this is probably only a temporary fix. The drug treatment would have to be repeated for life. The brain might already be damaged in these people, so maybe it's not. You can't undo the damage that's already happened. Let me just pause there so you can read those different things if you want to. And then let's continue. Okay, this is a tricky one. This is a suggest, okay? But you wouldn't be expected to know about the blood-brain barrier, so I don't, that's probably not on the mark scheme. But rather than taking a pill, it does give you a clue. So if you take a pill, you eat it, you swallow it, it potentially could be digested. So I think that's maybe what you need to go with. Taking a pill, potentially you could talk about stomach acid. Maybe the pH of the stomach acid damaging the drug. Maybe you could talk about enzymes digesting the drug, and I don't think talking about the blood-brain barrier would be on the mark scheme. There are going to be a few different things because it's a suggest, so they'll give you more than 2 options, probably 4 or something. Let's have a look. So those are the same one. So drug not destroyed by acid and drug not digested by enzymes. That's the same mark point. Actually, they kind of did want you to know something about the blood-brain barrier. So it's injected, which means it will directly reach the brain, and the cerebral spinal fluid bathes the brain. You don't need to know about the blood-brain barrier specifically, but this is a direct way of getting the drug to its point of action. But just, for your own interest, the blood-brain barrier is a tricky thing that doctors and pharmacologists need to get around if trying to treat the brain. Suggest and explain one way epigenetics may affect the age when symptoms of Huntington's disease start. We've already done some thinking about this, so epigenetics might affect the level of transcription and translation of the Huntington gene, and then I'd go on to talk about, for example, if the DNA in the gene is methylated, this will reduce transcription and translation of the gene, or if histone proteins in that gene are acetylated, is going to increase transcription and translation of the gene, producing higher quantities of Huntington, which would probably cause an earlier onset of symptoms. Yeah, that's exactly what we want. So if we talk about methylation, it inhibits or prevents transcription, or we could talk about the other thing, increased acetylation of histones stimulates allow transcription. In both cases, you can kind of talk about it from an increased perspective or a decrease, like more methylation inhibits, less methylation stimulates transcription, or you could talk about more acetylation promotes transcription and less acetylation inhibits transcription. Okay, so that's the end of that paper. I hope you found that useful. Best of luck with your revision in the coming weeks. Bye-bye.